Hello, students. This video is going to look at some of the processes that cause evolution to occur randomly, which is kind of a strange concept, but it does happen. And we'll sort of look at this from a statistical perspective in a certain sense, and we'll also look at this from a perspective concerning how this actually plays out in some naturally occurring populations. I think it is good for us to review again what evolution is and how it is that it occurs. We know that evolution is the change in frequency of inherited characteristics that occurs within populations from one generation to the next. This is sort of our working definition of what evolution is in a biological sense. And then we can talk about what causes evolution to happen. We know things like mutations, things like selection, either natural selection or sexual selection, things like genetic drift, or things like migration and gene flow. And genetic drift specifically is something that we're going to take a look at here. And so, what exactly is this thing called genetic drift? Well, genetic drift, simply put, is a change in allele frequencies that happens due to random chance alone. And we could sort of think of that as something being analogous to these five jars here that contain two different colors of marbles. And let's think of these two different colors of marbles as representing two different alleles that occur within this population. We have red alleles and blue alleles. And we could sort of think of this progression as representing time. And so we start off with having a jar that has about 50% red marbles and 50% blue marbles. And then at some later point in time, there is a higher frequency of blue marbles. And at a later point in time, an even higher frequency of blue marbles. And then maybe the frequency of red marbles is getting shuffled around. But ultimately, we end up, after 5 iterations of this example, having a jar that only contains blue marbles. And so these different jars are analogous to a population's allele frequencies changing from one generation to the next, and they're changing at random. We could look at this in a more quantitative sense. Basically, we could see uh, generations on this x-axis here going from 0 to 100, and the frequency of some allele we'll call p on the y-axis. And through this process of random genetic drift, what we see is an allele frequency that starts off at 50% at this initial point in time, and then it goes down, then it comes back up, and it goes down, and it goes back up, and it goes down, and it comes back up, and it goes down, and it goes back up and down and up and down and up and down and up a whole bunch. There are all these random fluctuations in the frequency of this allele P that have nothing to do with any of the other evolutionary forces. So we're saying if we put our population in a box or in a jar and we removed all of the other evolutionary forces, so we're saying there's going to be no mutation, there will be no selection, and there will be no gene flow, we would still observe allele frequencies changing over time, and this change, in fact, is entirely random. Now, how does this happen? Well, basically, genetic drift is something we call neutral evolution. And neutral evolution is a change in gene frequencies that occurs within populations which are completely unrelated to fitness. And a good way to think about this is in terms of what we call the coin flip analogy. We know we can flip a coin and we can either get heads or tails, and we seem to sort of have a 50% chance of getting heads or a 50% chance of getting tails. So if we think of these two alleles that a population carries, P and Q, as sort of being analogous to heads or tails of a coin, either allele sort of has an equal chance of becoming involved in fertilization. Each allele sort of has a 50-50 chance of being involved in fertilization. So if we imagine we have 3 potential genotypes composed of these alleles, we could have a homozygous genotype with P, we could have a heterozygous genotype that has both P and Q, and we could have a homozygous genotype composed of just Q. If there is random mating in the population, then these alleles will be pairing up at sort of random frequencies, and that kind of gives them an equal chance of being passed on into gametes and being involved ultimately in fertilization. So although we might know that we have these 3 different genotypes within a population, really we don't know which gametes are going to be the ones that ultimately become involved in fertilization. It could be one copy of P and one copy of Q. It could be two copies of P. It could be two copies of Q. And so really, even though we know that natural selection has influence on the frequency of alleles that are getting passed from one generation to the next, any two of these alleles really do have an equal chance at being involved in fertilization. So this is very much like flipping a coin. It is very much a random process. And so this thinking about random evolution is really something that specifically comes out of the neutral theory of molecular evolution. And this is something that was first formally proposed by the Japanese biologist Moto Kimura back in the late 1960s. Now, Kimura kind of was taking some ideas from folks who were part of the modern evolutionary synthesis of the early 1920s. People like Sewell Wright recognized that genetic drift was a thing, but people like these earlier biologists didn't quite recognize the importance of it. But basically, Kimura came up with this idea that probably most evolutionary outcomes really do have a molecular basis. So there is certainly a genetic basis dictating how gene frequencies are changing over time. Kimura postulated that yes, there are deleterious mutations, and most likely natural selection is going to be the force that removes these mutations from populations, and that also most non-deleterious mutations are neutral. That is, they're not beneficial. And we sort of know this from previous examples we've seen where most of the time mutations don't really do anything. They're silent mutations. Less frequently, mutations are not silent and they are harmful if they are not silent. And then even less frequently than that, there are non-silent mutations that turn out to be beneficial. And then also another tenet of this theory is that mutations can reach fixation by chance rather than via positive selection. And so one of the key ultimate take-home messages of the neutral theory of molecular evolution is that there is a large amount of genetic variation amongst many, many species, all species perhaps, whose destiny is really largely determined by random genetic drift. And I would point out that when we're talking about the neutral theory, we are in some ways often going to draw on examples of sexually reproducing populations. For example, when we're talking about this coin flip analogy and we're talking about different genotypes, we're talking about populations that are reproducing sexually. Now, this idea that Kimura proposed sort of led off on the heels of this thing we call the selectionist and neutralist debate. And basically, there were folks in evolutionary biology during the 20th century who sort of thought that evolutionary processes were really almost entirely dictated by natural selection. And there were other folks who thought, no, evolutionary outcomes, evolutionary processes are really more largely dictated by neutral evolution, things like genetic drift and mutation. And so some of the main ideas coming from these two different camps of evolutionary thinking during the 20th century. If we're to look at the selectionist perspective, uh, selectionists basically thought that balancing selection was the force that really maintained genetic variation within populations, and that probably natural populations tend to have really, really high genetic diversity as a result of this. In contrast, the neutralists kind of thought, maybe no, maybe genetic variation depends more upon mutation rates and population sizes, and that maybe natural populations kind of tend to really have low genetic diversity. And this is probably because selection's keeping wild type genes fixed, it's favoring beneficial genes, it's removing non-beneficial genes, and only occasionally do new mutations arise in populations that are either beneficial or harmful, such as to say mutations are relatively transient. So in short, selectionist perspective sort of postulates that most evolution is happening because of natural selection, and the neutralist perspective sort of postulates that most evolution is happening because of random mutations and random genetic drift. So we mentioned population size as being something important in the neutralist perspective. And as it turns out, the influence of genetic drift on allele frequencies within populations very, very much does depend upon population size. And so we can look at these different computer simulation models, and each of these computer simulation models involves multiple runs, which are represented by the different colors. And each of these model runs are attempting to simulate genetic drift under situations where there are different population sizes. So we have here a population size of 500,000. We have here a population size of 50,000, here 5,000, here 500, 50, and just 5. And in each of these simulations, the allele frequency of P here is starting off at 0.5. And in each of these simulations, we're looking at how the frequency of that allele is changing over the course of 500 generations on each of these different x-axes. And what we can see is something sort of interesting. When the population size is relatively large, the fluctuation in allele frequency caused by drift is very, very minimal. In this case, where the population size is 500,000, the frequency of the allele P never really gets too far away from where it started at 0.5. When the population size is a bit smaller, say 50,000, we start to see more variance in how, how this allele frequency is fluctuating. But it's not really fluctuating away from 0.5 a whole lot. It might get as high as 0.6. It might get as low as 0.4 in different model runs over time, but it really isn't fluctuating a whole lot. But what happens when the population size starts to get smaller and smaller and smaller? We're seeing tremendous amounts of fluctuation, particularly when the population size gets to be around 500 or less, to the point where the allele frequency, because of drift, could become as high as 0.8 or as low as 0.2, or even could find its way all the way to 0. Or in even more extreme cases, it could find its way to 1, where it could be fixed when population sizes are really small. And so I know this is kind of strange to comprehend, but the idea is very much, again, analogous to flipping a coin. We could think about the outcome of flipping a coin 500,000 times. If you flipped a coin that many times, chances are you're going to get really close to having heads and 50% of the time and tails 50% of the time. If you flipped a coin 50,000 times, you'd get pretty close to having having heads 50% of the time and tails 50% of the time, but maybe it might turn out that you'd get heads 60% of the time and tails 40% of the time, or vice versa. There's a higher frequency of a higher chance of not getting this 50-50 heads to tails ratio because you haven't flipped the coin enough times. Let's look at another example where you flip the coin 5,000 times. And now flipping the coin 5,000 times, it's possible to get 50-50, but now there's even a higher chance that you could get 70% heads and 30% tails, or vice versa. Flip the coin 50 times, and now it's possible that you could get 100% heads or 100% tails. And so that's kind of what's going on in these different simulations of how genetic drift is operating. It is very, very much probabilistic. And so the key message here is that as population sizes become smaller and smaller and smaller and smaller, the higher the chance that any two of these alleles really could be involved in fertilization at an unequal proportion that we would expect to be 50-50. This has some interesting consequences because once the population size starts to get kind of small, like less than 500, we see a situation where, simply due to random genetic drift, random chances of any two alleles being involved in fertilization, one of these alleles could be lost from the population altogether, or one of these alleles could also become fixed in the population altogether, which is not too different than what we saw under positive selection for very, very beneficial alleles. And so genetic drift has the ability to drive alleles to either loss or fixation, particularly when population sizes are pretty small. Now, again, these are computer simulations. These are sort of like statistical models, but actual real laboratory experiments have yielded quite similar results. In the 20th century, scientists work a lot with the fruit fly Drosophila, and they have raised Drosophila in controlled conditions and watched how allele frequencies in Drosophila changed, and altered the population size of these experimental Drosophila populations, controlling for the effects of natural selection, controlling for gene flow, attempting to control for mutation. And it turns out that this sort of process actually does happen in nature. So although the way we understand genetic drift is very theoretical, again, very statistical, we know that in fact there's evidence to show that it does happen naturally. Now, probably not surprisingly, the amount of time that it takes for a particular allele to either reach loss or reach fixation because of genetic drift, it depends upon population size, which we saw. But as the population sizes get smaller and smaller, the time for an allele to become lost or fixed starts to become lower. And so we could look at these three examples, sort of similar to what we just seen, with population size of 500, a population size of 50, or a population size of 5, 500 generations on each x-axis, and the frequency of some allele p on the y-axis. And what we see is that when the population size is 500, it could take as long as about 500 generations for an allele to be either lost or fixed. But when the population size is smaller, say like 50, then the number of generations it could take for an allele to be lost or fixed is something like 100 generations. And if the population size was only 5, the number of generations it would take for an allele to be lost or fixed might be as low as just 20 generations. And so the key message here, again, is that the influence of genetic drift on populations very, very much depends upon how large these populations are. Large populations are not as likely to be influenced by the effects of genetic drift as small populations. These random fluctuations in very large populations don't really lead to significant changes in allele frequencies, but in smaller populations, they do. So then we might start asking questions about, why does all of this matter? Okay, obviously genetic drift is influencing evolution, but why does this whole process of it affecting allele frequencies with changing population sizes matter? And as it turns out, genetic drift really can have a pretty significant influence on a population's genetic diversity, or what we might call a population's heterozygosity. And heterozygosity is a fancy term that represents a measure of genetic diversity, which we could quantify under the assumptions of Hardy-Weinberg equilibrium as being 2 times the frequency of the allele P times the frequency of the allele Q. This also could be simply quantified as the product of P and Q. Basically, if P is equal to 0.5, then Q is going to be equal to 0.5, and that is the highest amount of genetic diversity that a population can have at a particular gene locus. And this really comes back to this idea of balancing selection. Remember, under this model of balancing selection, there is a situation where the heterozygotes in a population have the highest fitness when compared to either of the homozygous genotypes. And so we know that this kind of helps populations deal with different aspects of the environment that could change. For example, things like resource availability, or things like responding to different chemical gradients, or things like responding to changes in climate, like we saw with Darwin's finches, or things like changes in physiography, which basically is a term that describes physical aspects of the environment, such as mountain building and river forming, or things like predation and parasitism, or things like disease. Populations with higher genetic diversity seem to have a higher chance at being able to survive changes in all of these factors over time compared to populations that don't have very high genetic diversity. And so when genetic drift starts to cause these random fluctuations in allele frequency over time, that leads to a situation where there is potentially a loss of genetic diversity, and particularly small populations are susceptible to this loss. And we know that low genetic diversity within populations can lead to this thing that we call inbreeding depression. And inbreeding depression is basically a situation where populations are composed of individuals who are so closely related to one another that they're leading to a higher frequency of homozygous genotypes being passed on from one generation to the next compared to heterozygous genotypes. And this is decreasing genetic diversity within populations. Sometimes we have these scenarios where populations are pretty stable and they have a pretty high amount of genetic variation, but then something causes the population size to crash, maybe a disaster, for example. And even though a population might recover from this disaster and start to increase in size again, what we see is that the genetic variation that the change in the change in genetic variation isn't increasing again. And so while this population size had a lot of genetic variation originally, population size crashed along with its variation because of the effects of random genetic drift. In part because of the effects of random genetic drift, it's going to be hard for this population to regain genetic variation again. And we can see examples of this thing we call inbreeding depression in these catch flies belong to the genus Silene. These are little wildflowers. But basically, the idea behind this situation where, let's say, there's a population who's lost a lot of genetic variation, and so individuals who are pretty closely related are mating with each other, is that it leads to inbreeding. And the level of inbreeding can sort of be quantified on this x-axis here. And we can look at fecundity or fitness on the y-axis here, which we might call germination success in the context of plants. That is, how many of these plants are successfully producing offspring that are going to go on to the next generation. And what we basically see is that when the level of inbreeding is basically zero, there's really, really high fitness in a population. This is basically a case where individuals who aren't very closely related to each other are involved in mating. But when individuals who are a little bit more closely related to each other are involved in mating, we have a little bit higher inbreeding, the level of germination success or fitness is going down and going down and going down to the point where when this population of, let's say, our catch flies, for example, is composed of a lot of closely related individuals who are sharing genes, forming offspring in the next generation, the fitness is pretty low in those populations. And this is something that, for one, can increase the chances of these so-called deleterious alleles pairing up. So sometimes homozygous combinations of alleles are very harmful. And this also makes populations a lot more, again, a lot more susceptible to environmental changes, as we were discussing under the context of balancing selection. And this often really is something that's associated with decreased fertility in populations. So why are we talking about this? Because genetic drift can start to cause allele frequencies to get out of hand in really small populations. And this can lead them towards something called inbreeding depression. So how can populations sort of work their way out of this? Well, the most obvious answer might be, well, maybe some new mutations could arise within the population that can restore diversity. And as it turns out, that doesn't seem to be in a very realistic or very effective situation at all. And this different computer simulation we're looking at here illustrates that scenario. So again, we have time and generations here on the x-axis, 100 generations. And we basically have heterozygosity here on the y-axis, or genetic diversity. And at some initial point in time, this population has a certain amount of genetic diversity. And we're going to assume that this is a very small population. And thus, it's going to really be susceptible to the effects of genetic drift. And if we were to model this under different situations where there were different mutation rates, we would see a different pattern of change to genetic diversity over time. And basically what we're seeing here is a model where we have these different mutation rates, a mutation rate of 0.1, a mutation rate of 0.01, mutation rate of 0.001, mutation rate of 0.0001, and a mutation rate where there's no mutations at all. And as it turns out, if the mutation rate is as high as, say, 10% mutations per generation, genetic diversity is going to be maintained in this population over 100 generations. If the mutation rate's a little bit lower, say 1% per generation, then we're going to see genetic diversity being lost, albeit a lot more slowly. But these are completely unrealistic mutation rates, especially for beneficial mutations. Instead, these lower values are the more realistic mutation rates, things like tenths and hundredths of percents of mutations happening per generation. And even with these realistic rates, it's projected that genetic diversity is still going to continue to decrease and decrease and decrease in small populations because of the effects of genetic drift. And ultimately, we might expect, depending on the population size, that eventually these one of the two alleles, P or Q, is either going to be fixed or lost, which basically leads to a genetic diversity or a heterozygosity value of 0, as if we might think about our simple calculation of what a genetic diversity is: P times Q, 2 times P times Q, however we want to quantify it. If P is equal to 1, then Q is going to be equal to 0. Vice versa, genetic diversity will be equal to 0. So new mutations are not likely to be very effective at restoring losses of genetic diversity in small populations. So we could start asking questions about, well, how else might genetic diversity be restored in small populations? Because we know this doesn't happen all the time. Well, as it turns out, gene flow is the most effective way small populations suffering from the effects of genetic drift, suffering from losses of genetic diversity. Gene flow is one way that populations can regain genetic diversity. And so we have a very sort of similar example here, a similar computer simulation. We have time again, 100 generations on the x-axis, and a measure of genetic diversity on the y-axis, which is the same measure of genetic diversity we saw on this y-axis. But instead of looking at mutation rates, we're looking at migration rates. That is, how many new immigrants enter a population per generation. And so we can start off with these different model runs again, and we start off with a certain amount of genetic diversity in a small population. And if this population hypothetically has as few as just 5 immigrants per generation, they will maintain genetic diversity pretty well. Even if there are only just 2 or 1 immigrants per generation, they're not going to lose a substantial amount of genetic diversity because immigrants are bringing new alleles or different alleles into this small population and mixing up the gene pool, causing genetic diversity to be maintained. If instead gene flow, the rates of gene flow, the rates of immigration are low, say really on average half an individual immigrant per generation, or even a smaller percentage of immigrants per generation, or no immigrants per generation, then we'll see a situation where genetic diversity cannot be restored. But the point is that migration is very effective at mixing up the allele frequencies within populations, again, and increasing their genetic diversity. And just to reiterate, 5 immigrants is a pretty small number, even 2 or 1. These are small numbers. So just a little bit of gene flow can make a big difference and really, really benefit small populations suffering from the effects of genetic drift and suffering from the effects of losses of genetic diversity. So we have talked quite a bit about population size here, and I think what's important for us to point out is that really when we're talking about population size in the context of genetic drift, we're talking about something called effective population size. And this is a sort of more pragmatic measure of breeding success. And so effective population size is not the same as census population size, which includes all of the individuals in a population. Instead, effective population size only accounts for the individuals that are breeding. And specifically, it accounts for the different numbers of breeding females and breeding males. And so again, we're very much talking about a population that reproduces sexually. And there are some different ways this could be calculated, but basically we could look at this as being a measure that is quantified as 4 times the product of breeding females, or the number of breeding females and the number of breeding males, divided by the sum of the number of breeding females and breeding males. And this number kind of points more accurately at how many gametes are getting passed on to the next generation. And so this is kind of an important way to look at the fact that if we're interested in how allele frequencies are changing, why will we consider population sizes with respect to individuals who aren't contributing alleles to the next generation? And we can ask, well, why not? Why is it that not all individuals could be contributing alleles to the next generation? Why not are all why are not why are all individuals not involved in reproduction? And there are a few explanations for this. For example, some individuals might not have reached reproductive maturity yet, and so they simply just can't reproduce. In contrast, some individuals may have surpassed reproductive maturity and are no longer involved in mating. Some individuals just might not be able to find mates, even if they are reproductively mature. They're just not successful at being able to reproduce. And so, for these different reasons, not all individuals in populations are reproducing, and so when we talk about population size, particularly in the context of genetic drift, we are talking about effective population size, particularly with respect to those individuals who are breeding in each generation. And as you might imagine, census population size is almost always going to be much, much larger than effective population. Now, this kind of gets us at this understanding of, okay, well, we know genetic drift is evolution at random, and we know genetic drift seems to really depend upon population size, or rather, the effect of genetic drift seems to depend upon population size. That is to say, the effect of genetic drift increases as population size decreases. And then the big question from an evolutionary perspective is, how does genetic drift interact with natural selection? And what we see is that genetic drift actually could overcome beneficial alleles that are under strong positive selection if population sizes are too small. And so uh, today, evolutionary biologists basically quantify the strength of genetic drift as the inverse of effective population size. And we know that the strength of natural selection is quantified via this thing we call a selection coefficient. And so then we could postulate some scenarios where, well, if the selection coefficient is much, much greater than the inverse of effective population size, then the strength of selection is going to be greater than the selection of drift. If the selection coefficient is sort of the same as the inverse of effective population size, then the strength of selection would be expected to be pretty much the same as the strength of drift. And then, sort of in contrast to the first example, if the strength of selection is much, much smaller than the inverse of effective population size, then selection will not be nearly as strong as drift. And so we can sort of look at this in some other computer simulations where we have an allele, we'll call it P, and this allele is under very, very strong positive selection. The selection coefficient is 0.01. That's pretty strong positive selection. And we have, say, 500 generations on each of these x-axes, and each different graph represents different population sizes. Population size of 5,000, 500, 50, or just 5. And what we can see is that when population size is equal to 5,000, the inverse of the inverse of that, or let's just say the effective population size is 5,000. The inverse of that is equal to 0.0002. That's a lot smaller than 0.01. Thus, we would predict that selection is still going to operate operate upon this beneficial allele, allowing it to reach fixation over some period of time, despite little bits of fluctuation in allele frequency due to drift. And again, as we saw in previous computer models, these are just different model runs. Each of these colors in these graphs just represent different model runs. And then if the population size starts to get smaller, say the population size is 500, or the effective population size is 500, we see quite a bit more fluctuation in allele frequency caused by drift. But again, the inverse of effective population size in this case, 1 divided by 50, is equal to 0.002. That's still smaller than the selection coefficient. So positive selection is still favoring this beneficial allele and ultimately leading it towards fixation. Once the population size starts to get smaller, now the effective population size, say 50, 1 divided by 50, or the inverse of this, is equal to 0.02. The strength of drift now is stronger than the strength of selection. And even though this beneficial allele is being favored by positive selection, and it might still get fixed under some of these simulations, it also could be lost simply at random because of drift, because the strength of drift is now overcoming the strength of selection because the population size, the effective population size, is too small. And even a more extreme example could be with a population size of 5, where the strength of drift is now much, much stronger than the strength of selection. And there's a much, much higher chance that a beneficial allele could be lost from this population simply because of chance, despite the fact that natural selection was favored. So this is kind of a simple breakdown of our understanding of the influence of genetic drift on populations. It causes evolutionary changes, but the magnitude of change that drift can have on populations, again, very, very much depends upon the size of the population. Now, there's another way that we can see random fluctuations in genotype and allele frequencies occur within populations that is quite similar to genetic drift, but it's happening in a much different way. And one of those is via something we call a genetic bottleneck. And genetic bottlenecks basically can cause allele frequencies to become rearranged at random due to things like disasters or disturbance events that populations might experience. The analogy in this situation could be this bottle we have full of all these different marbles, and these marbles come in 3 different colors. And so we can think of these 3 colors as representing 3 different genotypes. We have blue genotypes, black genotypes, and yellow genotypes. And the frequency of these genotypes makes up the gene pool in the bottle. And then if we were to take our hypothetical bottle and pour out some of these marbles into a glass, not all of them would get out because a lot of them would get trapped up at the neck of the bottle, sort of like a traffic jam. And so whatever the frequency of genotypes was at this initial point in time, once we get enough, we're able to get some of these marbles out of the bottle into the glass, we would see that the genotype frequencies would have changed at random. Let's just assume we had equal proportions of black and blue and yellow genotypes, black and blue and yellow marbles in the bottle originally. Well, the frequency of those marbles in the glass is not going to be the same necessarily. It's possible by chance that it could be, but there's also a very high chance that these genotype frequencies or the frequency of these marble colors are going to be rearranged at random. And that's what this cartoon is illustrating, a situation where we go from having, again, equal frequencies of these different colored marbles to having a completely different frequency of these marbles. And in fact, in this example, the yellow marbles have been lost from the population altogether. So it's very much sort of a similar thing that we see going on with genetic drift, where random changes to genotype and allele frequencies happen. These bottleneck events, again, generally occur following population crashes. And population crashes could happen because of disturbance events in natural populations, things like wildfires, things like floods, just to name a few examples, things like even wind disturbance, things like human disturbance or anthropogenic disturbance, or even things like diseases. Populations are quite susceptible to disease often, and that can wipe out large numbers of individuals in populations, causing these bottleneck events. Now, basically, we could look at this graphical example. Again, time on the x-axis, population size on the y-axis. Population size is fine. It's stable for some period of time. And then we have this population crash following a bottleneck event. And so population size declines a lot. And then there are one of 2 possible outcomes in this situation. Either the population is able to recover from the disturbance event or the disease, the bottleneck event, or the population cannot recover because its size is so small and its genetic diversity is so low that the population might ultimately just go extinct. So we can actually take a look at a real example of this in nature, um, which is something that happened to the northern elephant seals. And I believe it was during the late the late 20th century, I'm sorry, the late 19th century, that northern elephant seals, which occur in the northern Pacific Ocean, were nearly hunted to extinction. And this hunting pressure led them to experience a bottleneck event where their population sizes crashed because so many individuals were being hunted. And as a result, they lost a tremendous amount of genetic diversity. We can compare the genetic diversity of the northern elephant seal today to the genetic diversity of their sister taxa, the southern elephant seal, sister taxon, the southern elephant seal. And that's what's sort of illustrated here on this graph. So basically, we're looking at some measure of genetic diversity here on this x-axis, and we're comparing northern elephant seals in blue to southern elephant seals in orange. And we can see that northern elephant seals have basically no genetic diversity at the particular loci that were being studied. And in fact, most of the loci in the northern elephant seal lack polymorphism. That is, most of them are homozygous. And so here's a population that experienced lots of hunting, underwent a bottleneck event, but was able to recover. But despite its recovery in size, population size, it has not recovered in terms of genetic diversity. And this is sort of a dangerous situation for the northern elephant seal, because although this species is doing fine today, it is extremely susceptible to any particular type of environmental change that might come along, such as climate change, or such as changes to the habitat where they occur within the ocean, or diseases, for example. Another interesting caveat to the story of the northern elephant seal is the fact that their populations tend to be dominated by just a few mating males who are very, very dominant. We call these guys beach masters. And so this is an interesting situation that is affecting the population size, or really has an influence on the effective population size of the northern elephant seal. And so even if we have, hypothetically, let's say a population of 100 northern elephant seals, 50 males and 50 females, maybe only 2 of those males are actually involved in mating with the 50 different females. This is a realistic scenario with this species. And so even though their population size at the census level is 100, the effective population size, by virtue of only having 2 breeding males, is much, 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 much lower. And this is sort of exacerbating losses of genetic diversity even more as these populations are reproducing again and again. So bottleneck events can have some interesting implications. Another similar example of something like this is what we call the founder effect. And rather than populations having their size altered by some sort of disaster or some sort of disease, in this case, we have a large population within which a small subset goes on to colonize new ground. And so in this example here, we have these plants that occur on some mainland environment, and this population of plants in the mainland environment is composed of some different allele and genotype frequencies associated with some different characteristics that these plants have. And it turns out that these plants produce fruits, which are a favorite food of some bird species. And so this bird species will feed on the fruits of these plants. And this bird species is very mobile, and it can fly out to an offshore island and defecate out these seeds. And the seeds can survive the journey and germinate and form a new population of these plants on this island. But what we see here is something interesting. The allele frequencies on the mainland are not the same as the allele frequencies that end up happening on the island. And this is happening at random. So imagine in this hypothetical cartoon, the allele frequencies on the mainland are something like big R is equal to 0.3 and little r is equal to 0.7. And we could calculate that looking at all of our different plants here on the mainland. But by virtue of this bird species randomly picking fruits off of these plants, flying out to this island and depositing these seeds on the island, the allele frequencies have completely changed, and they have changed at random. Now, the allele frequencies on the island are big R equal to 0.6 and little r equal to 0.4. We have a change in allele frequencies going from one environment to the next, going probably from one generation to the next. This is evolution by definition being caused at random via something we call the founder effect. We have a real example of this with this bird comes from Australia. And so in this case, we're just going to talk about the bird, not plants. But basically, this is something called a silver eye. And these silver eyes originally occurred on the Australian mainland. But they made a series of colonization events from the Australian mainland to Tasmania, to New Zealand, and ultimately out to this place called Norfolk Island. And so this graph here represents these different environments on the x-axis. ML is the mainland, T is Tanzania, SI is the South New Zealand island, et cetera, all the way out to Norfolk Island. And on the y-axis of this graph is basically a measure of genetic diversity. And what we see from this graph is that there was a certain amount of genetic diversity that these silver eye populations have on the Australian mainland. But as these smaller subsets of this population went on to colonize Tasmania and New Zealand and ultimately Norfolk Island, this genetic diversity was being lost. And this was happening as a result of the founder event, which is not terribly different than the bottleneck event. And so this has some interesting implications. Can these different silver eye populations deal with these different environments that they're colonizing, despite the fact that they have kind of successively lower and lower genetic diversity, or not? Will these things be able to come back and breed with one another? That's kind of another interesting question. And so we see this real life situation where because of this thing called founder effect, as populations go on, small subsets of populations go on to colonize new ground again and again and again and again. They're losing genetic diversity as their allele frequencies are being mixed up at random and as their population sizes are becoming probably, I would assume, smaller and smaller and smaller and smaller. And so this kind of situation where we see sort of differences in spatial distribution within populations and probably differences in the genetic diversity among different subsets of this population tends to play out in an interesting way. And it causes something that we call isolation by distance. And this is basically the concept that if a species is broken up into a bunch of different constituent populations, those that are spatially distributed closest to one another will probably have the highest chances of breeding with one another. And so let's just consider this cartoon. We have 5 populations, A, B, C, D, and E, distributed across some geographic space. Those that are in the closest proximity are probably going to have the highest chances of dispersing and exchanging genes with one another. So for example, population A and population B, they're closest together in space. They're probably going to have the highest chances of exchanging genes. Likewise, population B and population C, pretty close together. They're going to have the highest chances of exchanging genes compared to other populations, et cetera, et cetera. Whereas maybe population A and population C, well, they don't have as high of a chance of exchanging genes as A does with B, or as I should say, A doesn't have as high of a chance of exchanging genes with C as it does with B, but it certainly has a higher chance of exchanging genes with C than it does with D. And likewise, it has a much lower chance of ever getting to exchange genes with E. And we could say this same thing about all the different relationships amongst all of these populations. And this is leading to some interesting patterns. Individuals who are closest, or individuals within populations who are most geographically closest to one another are more likely to be genetically similar. And we can see a real example of that with the Florida scrub jay. So basically, this little map here on the right illustrates a bunch of habitat patches occupied mostly by Florida scrub jays. And the the colors in the different plots are basically representing the genetic differences between them. And so individuals in populations that are more blue are more closely related, and individuals in these populations that are more red are more closely related. And what we can see across this geographic space is that the individuals who are most closely related to one another also tend to occur more closely to one another than to other individuals in populations that they are more distantly related from. And this, again, is what we would call isolation by distance, driving differences in gene frequencies amongst different subsets of this larger Florida scrub jay population as a whole. And so there are distinct genetic differences being distributed across geographic space. Now, this leads to this question, are the patterns that we see here due to neutral evolution or due to natural selection? That is to say, is the distribution of genetic diversity in this landscape occurring completely at random because of genetic drift? Or is it that individuals here more associated with this blue color are locally adapted to the environment here, and individuals down here with more reddish colors are locally adapted to this environment, perhaps because the forces of natural selection operating here are much different than the forces of natural selection operating here. Well, as it turns out in this example, this is actually a pretty small area. If you look at the scale bar, this is 2 kilometers represented on the scale bar. So we're looking at about 9 kilometers of space here. That's not a lot of geographic space within which forces of natural selection are likely to vary uh, in terms of their influence on this species. And so we're led to believe that isolation by distance is something that's probably really, really more being driven by neutral evolution. The differences amongst these populations genetically are really happening probably because of drift and probably because of the fact that birds that occur over here are more likely to breed with one another, and they're less likely to travel all the way over here to breed, and vice versa. The birds that occur over here are less likely to travel over here to breed. They're more likely just going to stay where they're at and breed with individuals that are closer to them. It takes energy to travel. What's the point in these birds traveling so much further to find mates when they could find mates really right in their own backyard? And this concept of isolation by distance really leads to another concept that we call genetic distance. And as it turns out, the genetic distance between populations is usually correlated with the geographic distance between them. And so one way we can look at this is by comparing two hypothetical populations, and we can look at the allele frequencies of each population and compare them. And in this example on the top, it turns out that the allele frequencies of these two populations are the same. And so we would say, well, the genetic distance between these populations is 0 because these two populations are genetically identical. In contrast, if we look at this sort of bottom example, we can compare a situation where the allele frequencies in these two populations are completely different. In this case, population 1 is dominated by allele A1 and population 2 is dominated by allele A2. And so we would say that the genetic distance between them is equal to 1, or they are 100% genetically different. And so we can sort of take this information. For example, if we looked at bighorn sheep in the California desert, that's where this data comes from, we can compare the genetic distance between populations to the geographic distance between them. And as it turns out, populations that occur further apart from one another do seem to share fewer and fewer alleles. There is a greater genetic distance between populations that also share greater geographic distance. And so we could look at relatively low genetic distance here, and that would be associated with relatively low geographic distance. These populations occur pretty close to each other, and it's not surprising that they're fairly genetically similar. We could look at higher levels of genetic distance where these populations over here occur pretty far from one another, and not surprisingly, they're a little bit genetically different from one another, or quite a bit genetically different from one another, about 45% genetic distance between populations that occur this far apart from one another. And so this sort of genetic distance model is very much consistent with this idea of isolation by distance. When populations are separated from one another in space, they tend to not share genes with each other as much as populations that are closer together in space. And this ends up leading to some important consequences down the road when we start talking about how populations might actually start to become differentiated from one another. So that's it for this lecture. I'll see you next time.